Okay, so welcome back to this next video in which we are discussing the detection of protein palmitoylation using tritiumated palmitic acid. Okay, right, so we've discussed the tritiumated palmitic acid that we're going to use, and we've discussed how it's going to be releasing beta radiation. Okay, so what we're going to now do is choose a cell line, and I've given you 4 examples, and we're going to transfect this cell, all of the cells that we've got of this type, with an extra copy of the gene for the protein that we are investigating for palmitoylation. Okay, and this is so that the so that the cell will be making a huge amount of this protein. Okay, so that we've got a lot of this protein to work with. Okay, right. So now what we're going to do is starve the cell for an hour, basically. Okay, so this is so that the cell will run out of normal palmitic acid, basically. And so when we add in our radioactively labeled palmitic acid, suddenly the cell will use that palmitic acid to add on to the protein A that it's been producing, basically. Okay, so the experimental procedure then, we've started with transfect the cells. Okay, so we're adding in an extra copy of the gene, so the cells will all be making much more of the protein. So transfect the cells is step 1. Okay, the next step is going to be to starve the cells, and we're going to do this for 1 hour. Okay, and basically you starve the cells in a medium that contains 2% dialyzed fetal bovine serum. Okay, so the medium is going to contain 2% dialyzed fetal bovine serum. Okay, right. And now once they have lasted for an hour, okay, so they've run out of palmitic acid, what you're then going to do is you're going to put them into a medium, you're going to add to their medium tritiumated palmitic acid. Now, you're going to add palmitic acid which has a certain level of radioactivity, basically. So, for 4 hours, what you're going to do at 37 degrees Celsius, and I should have also stated that the starvation process, which you do for an hour, that again will be at 37 degrees Celsius. Okay, so after you've starved them for an hour at 37 degrees Celsius, what you'll then do for 4 hours, again at 37 degrees Celsius, is that you will expose them now to a concentration of tritiumated palmitic acid, which results in you having a concentration of radiation of 25 to 100 microcuries per milliliter, basically. And this is going to be 9 at 10 tritiumated palmitic acid. So let me explain what this means. So basically, you are going to have a certain concentration of palmitic acid in the solution that you will be adding on to your cells and exposing them to for 4 hours. Now, the palmitic acid has a certain likelihood that it will disintegrate at any second, basically. Okay, so you will need a concentration of palmitic acid that results in a certain number of disintegrations per second, and curies is basically just another unit for disintegrations per second, but let me show you that in a moment. Okay, so you're going to add basically a certain concentration of 910 tritiumated palmitic acid, and this concentration will need to result in a certain level of radiation. So basically, curies, which is abbreviated to CI and is named after the 2 Curies, Marie Curie and her husband, okay, who did a lot of work with radioactive substances. Okay, so 1 Curie is basically equal to a certain number of disintegrations per second, and that number is 3.7 times 10 to the power of 10 disintegrations per second. Okay, so DPS, that should say, not disintegrations SP. DPS, disintegrations per second, okay, or decays per second. So this means decays per second. Okay, so basically this is 37 billion decays per second. So if your substance is 1 curie, then that means that you'll be getting 37 billion disintegrations off that every second. So you'll be getting 37 billion electrons coming off effectively. Okay, so what does this mean then? 25 to 100 microcuries. Well, firstly, a microcurie will be this divided by a million, basically. So a microcurie is going to be 3.7 times 10 to the power of 4 rather than 10 to the power of 10. So all I've done is divided by 10 to the power of 6 there. So this is going to be 37,000 decays per second, basically. So let's fix one of those numbers. So let's, for instance, just say that we want 25 microcuries per milliliter. And what that will mean is that we want 25 times this. So we'll take 25 times 37,000. And that will give us the number of decays per second. And you will want that number of decays within a milliliter. Okay, so 1 milliliter is 1 centimeter by 1 centimeter by 1 centimeter. So it's roughly a box around that size there. Okay, so in 1 centimeter squared of, cubed of the solution that contains palmitic acid, you want to be getting whatever 25 times 37,000 is. You want that number of decays coming off, and we'll try and work this out. So we can do this by multiplying by 100 and then dividing by 4. Okay, so if we multiply by 100, that will take us up to 3.7 million. If we divide by 4, that will take us down to 600,000. And then we'll have 100,000 divided by 4, which will go down to 25. So I think that comes out at 625,000 decays per second per milliliter. So off a milliliter of your solution, you want to be getting 625,000 decays every second. Okay, and from that you can work out what the concentration of palmitic acid, of tritiumated palmitic acid that you're going to need within this is. Okay, once you know how likely a single palmitic acid molecule is to decay in a certain second. Okay, so that's what that means. It's just telling you that you need a certain concentration of palmitic acid, basically, but it's not doing it in a particularly amenable form. It's telling you how much you need by telling you how much radiation you need to be emitting from that concentration every second. Okay, right. So you will expose these starved cells to this 25 to 100 microcuries per milliliter solution of 9,10 tritiumated palmitic acid. Okay, now that will occur for 4 hours. The next step is to rinse off the palmitic acid that you've put over the cells. Okay, and you do this with a solution that is known as STE fluid, capital STE fluid. Okay, so now the next step is to rinse the spare tritiumated palmitic acid off, basically, because you don't want that affecting your results. You want only the palmitic acid, you won't want only the tritiumated palmitic acid that is within the cells to remain on there. Okay, so what does STE fluid contain? Well, basically, its main component is sodium chloride. So STE fluid consists of 100 millimolar sodium chloride. Okay, so that's just a sodium ion bound to a chloride anion by a ionic bond. Okay, the next thing that you have within STE fluid is you're going to have 10 millimolar of a substance that is often just called tris. Okay, now what is tris? Well, let's discuss what tris is. So tris is short for tris hydroxymethyl aminomethane. So tris hydroxymethyl and then aminomethane. Okay, so let me show you the structure of tris hydroxymethyl aminomethane then. So basically what you have is a carbon atom with an amino group coming off it. Okay, so this is the aminomethane bit down here. Here's the amino group. Okay, so I'll highlight different bits and show you how the name reflects the structure. Okay, so the amino here refers to this amino group here. Okay, the methane refers to this carbon with the amino group coming off it here. Okay, now the other things that are coming off this carbon are the tris hydroxymethyl. So you're going to have 3 hydroxymethyl groups coming off it. Okay, so this basically consists of a methylene group, then with an alcohol group coming off it, like so. And you'll have 3 of those. So they're coming off in all directions, basically. So here's another methylene group with an alcohol group coming off it. And finally, another methylene group, then with one final alcohol group coming off it. So this is tris hydroxymethyl aminomethane. Okay, so you're going to have 10 millimolar of this substance, which is just often abbreviated to tris, in the STE fluid. Finally, another substance that you have in there, apart from obviously water, is 1 millimolar of a substance known as EDTA. Now, what does EDTA stand for? Well, it stands for ethylene diamine tetraacetic acid. Okay, so I think we better do this here. Okay, so EDTA is short for ethylene, and it's all one great big word. So ethylene is the E, the D is then for diamine, and I'm not going to be able to put this as one big word, but I'll do my best. Okay, then the T is for tetra, and now this should be adjoined onto the tetra, but never mind. Acetic acid. Okay, so the A is then for acetic acid. Okay, so ethylene diamine tetraacetic acid. So, what's the structure of this then? Well, basically, let's start off with the ethylene. Ethylene basically just means 2 methylene groups joined to one another. Okay, so here is an ethylene group. Then you need diamine, so that's telling us that we need 2 amino groups coming off. However, they're not pure amino groups. If they were pure amino groups, we'd be stuck then. It would just be ethylene diamine, okay? Or rather, 1,2-diaminoethane would probably be a better name for it. But instead, what you've got then is acetic acid groups coming off these nitrogens, basically. Instead of the normal hydrogens, instead you've got acetic acid groups, and this is why it's called tetraacetic acid. Okay, so acetic acid is the old name for ethanoic acid. So these groups that are coming off are 2-carbon carboxylic acid groups. Okay, so here's the ethylene diamine, here is an acetic acid group, and I've drawn the methylene group just as a CH2 group rather than as a carbon with 2 hydrogens coming off, just because otherwise it gets kind of crowded when you're drawing this structure. Okay, so again I'll show the methylene group just as CH2 there, and then you have a carboxylic acid group coming off here. Okay, so that's 2 acetic acid groups now. Let's put another one, well, another 2 over here. So here's another methylene group, and here's another carboxylic acid group over here. Okay, and then here's another acetic acid group here, CH2, and then a carboxylic acid group here. Okay, right. So that's ethylene diamine tetraacetic acid, EDTA. Okay, right, so those are the 3 things that you will have in the STE fluid. You'll have sodium chloride, 100 millimolars of that, 10 millimolar of tris, and 1 millimolar of EDTA. So it goes down nicely, dividing by 10 each time. Right, okay, so we've now rinsed off our solution containing the tritiumated palmitic acid. What we're now going to do is lyse our cells open. Okay, now, before we go on to talk any further, let's think about what this process of adding in the tritiumated palmitic acid has done. Okay, so our cells were starved. they had run out of palmitic acid. So when you suddenly started giving them this tritiumated palmitic acid, they were overjoyed. They said, fantastic, this is what we've been looking for. We've been synthesizing all this protein A because you've put an extra gene into us, okay? And we couldn't palmitoylate it, okay? So we're assuming, remember, that protein A is a protein which gets palmitoylated, okay? So we've got all these protein A molecules that are waiting to be palmitoylated. You've just given us this palmitic acid molecules. We don't care that they're tritiumated, so we're now going to stick them onto protein A molecules. So what's going to happen is that protein A, if it is a protein which has palmitoylation sites, is going to get these tritiumated palmitic acid molecules stuck onto it. Okay, so I'll just show, let's say it gets 3 of them added onto it, and I'll put these in the best radioactive colour that I have, and which is that. Yellow would probably be the best colour, but I'm not sure this will show up. Okay, so these are radioactive palmitic acid molecules. They have 2 tritium atoms in, remember. Okay, so now what we want to do is break the cells up, get this protein A, which has these tritiumated palmitic acid molecules, out, and we then want to somehow separate them from the rest of the gloop that we get, and somehow, well, then assay whether they're radioactive, basically. Okay, and that's what we'll continue in the next video.